This is a worm.
But not just any worm.
It's an arrow worm!
Unfortunately, Bill got caught up filming his volcano episode, so he asked for us to fill in for him today.
Bill Nye the Science Guy.
Brought to you by Biology 111.
Today, Bill asked us to talk to you about his favorite taxa, the Chaetognatha.
Chaetognatha.
No, see? It's Chaetognatha.
This is the Chaetognatha.
The Chaetognatha, also known as the
Arrow worm!
is a taxa made up of almost 200 species of planktonic worm-like organisms.
The arrow worm is formed of two phyla, the Phragmophora and the Aphragmophora.
Today we will be discussing the characteristics that define this taxa.
Here is a drawing of the arrow worm!
Defining characteristics of the Chaetognatha include its streamlined appearance and hooked set of jaws protruding laterally to the mouth. Arrowworms are made up of several paired lateral fins and a single tail fin. The female reproductive organs, or the ovaries, are found behind the mid-body mark, while the testes and the gonads are located in the tail. Chaetognatha have a bilateral symmetry, are segmented, and have fins or paired appendages which allow them to swim. However, they have a head which is indicative of cephalization. Arrowworms have a nervous system made up of a circumpharyngeal ganglionated ring and a digestive system that works by pushing food through the gut and eventually out a non-terminal anus, but no circulatory system. Lastly, they have more than 2 cell layers, tissues and organs, and a body cavity known as the coelomate. Phragmophora and Aphragmophora are 2 phyla within the Chaetognatha taxa. These phyla are differentiated by the presence or absence of the ventral transverse muscle bands. Transverse muscle bands are characteristic of Phragmophora, but are not found in Aphragmophora.
Back in the lab, we have been looking at the evolutionary origins of Chaetognatha, and we have built 2 phylogenetic trees. The first one is built off of the physical characteristics that Chaetognatha present. The second one is built off of the rRNA sequences of Chaetognatha and other taxa. In the first tree, Chaetognatha was shown to have a most common ancestor with Annelida. This is because their characteristics are very similar and they only share 3 differences in their characteristics, those being their appendages, their locomotion methods, and their circulatory system. In the genetic tree, Chaetognatha was shown to share a common ancestor with Arthropoda. This is due to the least amount of differences in the 2 DNAs. The 2 different placements of Chaetognatha in the phylogenetic trees are due to the fact that Arthropoda and Chaetognatha look very different, however, their DNA is more similar. As a result, in the first tree, the closest relative to the Chaetognatha was Annelida, and in the second tree, its closest relative is Arthropoda. Chaetognatha was discovered in the 18th century, but it has been traced back to the early Cambrian period. Scientists disagree on the placement of Chaetognatha in the phylogenetic trees. This is because arrowworms have characteristics of development that are classified as deuterostomes. However, genetically they are more similar to protostomes. Despite all of this, it is agreed upon that Chaetognatha are coelomates that develop from pseudocoelomates and acoelomates. We were unable to find a phylogenetic tree depicting Chaetognatha in relation to other taxa in literature. However, the phylogenetic tree that we made in lab about the characteristics of Chaetognatha show that Chaetognatha are more closely related to Annelida, which are protostomes. This matches the literature found that says that Chaetognatha are more closely related to protostomes. Thus, they are more closely related to Annelida.
Chaetognatha live in marine environments just like this, as serene environments in polar and tropical regions. Some live just above the deep ocean floor. They can also be found in midwater, neritic water, rock pools, or associated with certain ocean currents. Chaetognatha are carnivorous predators that eat crustaceans, mainly copepods, and some small fish. They feed by secreting substances that allow them to stick to their prey and attach their mouths and vegetables. Their prey is detected by the cilia on the worm, causing the head to dart forward and capture the prey using their hooks. Next, they inject their prey with a neurotoxin, tetrodotoxin, and swallow their prey whole. In order to catch prey, Chaetognatha need to be able to move. Airworms swim by contracting the muscles of their right and left sides alternately, which creates forward darting motions. They are hermaphroditic, meaning that they have both male and female sex organs. Fertilization in airworms occurs internally, and afterwards the eggs can be released into the water, on the seafloor, or brooded in pouches in the tail. Development occurs very quickly once the eggs have been fertilized, and typically they go from zygote to juvenile within 48 hours. The lifespan of airworms changes depending on the location where they live. Cold water airworms are expected to live 2 years, while tropical Chaetognathus have a much shorter lifespan of about 6 weeks. Although we may not see Chaetognathas, they do have an indirect relationship with humans. Chaetognathas can be hosts to parasitic organisms and can pass parasites onto their predators and up the food chain. This affects humans as many of the fish that we eat may have accumulated the parasites that the arrowworm hosted, and therefore the parasites may be transferred to us.
So that's it for this week's episode. We have to go get back to the, what do you call it?
Wait, I don't know.